Hello students, this is Dr. Faizan Mirza. In this video, I'm discussing with you the solved part of October-November 2025. This is paper 4, variant 1. Okay, so let's start. The first question here came for genetic trust. They have given to you a detail of a guinea pig. Here you can see, although not asked here, the name mentioned in italics is its actual name, or you can say the name with which we call the organism, the binomial nomenclature. The first name represents the genus. You know that whenever we are using the binomial nomenclature, the first name represents the genus and the second name represents the species. So and the common name is just guinea pig. So this is the guinea pig here. They have told to you that there are two genes that determine the length and color of the fur. So there are the two features. One is the length and the other is the color. And then they have mentioned to you that this gene, capital A, small a, is at locus, and then capital B, small b, is at another locus. They are both on separate autosomal chromosomes. So this is not a sex-linked condition. Then whenever you are given these kind of bullets, I ask you, or I let you know that in fact I encourage you that you should annotate it here on the paper together with when you are reading. For instance, they have mentioned you that allele capital A is for short fur. So capital A is for short fur, which means capital A dash is short fur. Capital A dash means that capital A with another capital A would be short fur. And similarly, if you fill this dash with a small a, again, capital A, small a is short fur. So A dash is short fur, small a is long fur. They have told you that A is dominant to A. The capital A is dominant. Then the allele capital B, the same goes for B, that black fur is B dash and chocolate fur is small b. Small b, B is dominant to small b. So I have just mentioned the phenotypes accordingly. Okay, so if the guinea pig will have at least one capital A, its fur will be short. If at least one capital B, its fur color will be black. The only condition for the fur to be long or chocolate means it must be small a for the fur to be long and small b for it to be chocolate. List all the possible genotypes of guinea pig with short black fur. Short black fur means that now you have to mention how the feature of both align together. So for short, we know that it should be capital A dash. So we will use the capital A dash Wali condition. So this is capital A dash, capital A dash, capital A dash, and this all four combination in which in these we have aligned them with both capital A's together, one capital A, one small a, one capital A, one small a, and one capital A, with all the possible combinations for the black fur. And black fur is capital B. Now, capital B is capital B dash. So this can also be of all the conditions that it can be capital B, or capital B, small b. So these are the four possible combinations of this particular feature. Then they are asking you how a test cross is carried out. So whenever we are carrying out a test cross, so this is something to be just kept in mind always whenever for a whichever cross. Whenever we are doing the this test cross, we always cross it with the homozygous recessive for both the traits, which means the purebred recessive or the small a, small b, in which both the traits are being there displayed as the recessive traits. And then we look at the offsprings in the next generation, whether they show the recessive trait or not. Because if they will show the recessive trait here, it means that this parent even passed on the small alleles, the recessive alleles, and the other parent also passed on the recessive allele. So the chances of an offspring with long fur and chocolate fur will be there if the other parent is also having the recessive alleles together with the dominant allele. So let's move on. Next, they are asking you to put a cross here and put a cross together here. A black guinea pig with long fur that was homozygous at both loci. So it's homozygous at both loci was crossed with chocolate guinea pig with short fur that was homozygous at both loci. So we know that this is homozygous at both loci. So with long fur, black with long fur, and homozygous means that it must have both the same letters, either the capital or the small. So this is the one, and then this is the chocolate guinea pig with short fur. So that will be capital A, small b, small b. And when they will form the gametes, we know that the gametes will have one letter of each. So we put the letters together in the circle. So these are the two gametes that they will form. The F1 offsprings of the cross had short fur. F1 offsprings were mated together to produce the F2 offspring. So when you cross them together, these are the offsprings that you get. When these are the offsprings that you get, they're asking you to cross the F1 offsprings together. So these are the F1 offsprings. It's simply a 9331 cross. So what will you do? We'll put together a cross with all the four possible gametes here on either side and start to cross them together. The eventual phenotype will come out to be 9331, 9 showing dominant feature, 3 showing dominant recessive feature, 3 showing recessive dominant, and 1 showing recessive recessive. You can just align the four type of features from the cross once you perform it. Some genes in the guinea pigs are structural genes, some are regulatory genes. Which ones are structural genes? Structural genes we know are the ones that add a functional protein to the cell, and regulatory genes are the ones that that control the transcription of another gene or expression of the other gene. So regulatory gene codes for the repressor protein or a transcription factor. And using these two, either of these, the regulatory gene will regulate the expression of the structural gene, and the structural gene they will then code for the functional protein such as an enzyme. Then they're asking you to just compare the structural features, one, and the functional difference between these two organisms. They have told you that this organism is a fungal pathogen, and this one is a leaf. So this is fungi, and this is plant. So this question, then this is second question. This deals with kingdom plant, and this deals with kingdom fungi. So you recall the feature of the fungi and plant, and then start to put the table together. Again, recalling the first word in the italics represents the genus. So these two are the genus of these organisms, and the second word represents the species. Okay, so the structural difference between the cell wall of fungi and plant is simply whether the cell wall is made up of chitin or cellulose. And then the structural difference could be that there is one nucleus in a plant cell, whereas fungi will have more than one nucleus, the multinucleate syncytium. And then fungi always lack chloroplast, whereas plant leaf will have chloroplast. Functional difference, how the function varies. So the function varies at heterotrophic or autotrophic nutrition. So I just made a spelling error there for autotrophic. But anyhow, so that's what you need to just mention the functional difference. Okay. Describe the principles by which the organisms such as these are classified by in the taxonomic hierarchy. So the taxonomic hierarchy that begins with domain, then kingdom, and it ends at species. How are these aligned? So these are placed in taxa. What are taxa? Taxa are simply our groups. These are the groups which are larger groups. They are divided into smaller groups. So domain is the larger group. It is divided into kingdom, which is a smaller group, and so on and so forth. The organism within one group shares similar feature with the other. That's why they are placed there. So initially, the organism is placed in one of the three domains. There are three domains: the bacteria, the eukarya, and the archaea. Both these organisms are in the domain eukarya. We know that because one is fungi, the other is plant, and they get further divided into kingdom, phylum, class, order, family, genus. You need to remember the hierarchy. And then Z maize is in the kingdom of plantae. This one is in the kingdom fungi. We just mentioned, and then we get our good 4 marks. Next, they are saying that this is the bar chart showing to you the when two varieties of maize were crossed together and F2 plants were grown, the percentage area of leaf damage caused by this particular fungal pathogen was measured. The arrows show the leaf damage of the two varieties. So the number of plants are shown with these bars, and these are the two varieties which are damaging. You can say the varieties of the maize which are given here, and then there you are saying how much percentage leaf was measured was damaged by this particular fungal pathogen. Explain how figure 2.1 can be used to determine the parent maize variety shows greatest resistance to infection by this fungal pathogen. So if we say the lag shows the percentage damage, it means that it is having a high resistance to infection, because if it number of plants are not getting infected, so this means they are resistant. SKB50. So where did we pick it? This is SKB50. Is showing only 22%, showing greater resistance to infection than CML 153, which is 58%. So where did we get these things? We are getting these things from these points only, and we accordingly just compute them in our answer. Okay. State the name of the type of continuous variation showed by the type of variation showed by maze F2. So this is continuous variation. And why continuous variation? Because this is a feature represented by bar charts. If these were not bar charts and there were gaps here, so it would have been discontinuous variation. Plus, there's a whole range. So when there's a range and there are intermediate phenotypes exist, so this is continuous variation. Explain the genetic basis of type of variation shown by maze F2 generation. Genetic basis. So what is the genetic basis of variation? They often ask you the genetic basis of variation or the environmental basis. So the genetic basis is this, that this is a continuous variation, yes. And then when the continuous variation is occurring, we know that there are multiple genes that are involved in controlling the single trait. In this case, the resistance to this fungal pathogen. These genes will show additive effect. The more the genes you have, the more resistant you will be. If an individual, or vice versa, if an individual has more dominant alleles of those genes, it would express stronger phenotype, and the reverse argument can be given there. Alleles of one gene will have a smaller effect on phenotype. For instance, if this thing is 22% resistant, so it's having more number of genes that are adding up together to affect the resistance, while the other one is you can say is being more affected by the infection because it's having the it's on the other end of the spectrum. Question 3 deals with two pigments taken from two chloroplast pigments taken from different sources. They are saying there are two type of methods. One is TLC, and the other in which the this chromatography was done, and the solvent was changed. And this is saying that this is here the if you see figure A and figure B, chromatograms represents that on this is the line of origin at which you place the spot of the chloroplast mixture and you place them in these solvents. And then the spots, they migrated up depending on the solubility. So we can see that xanthophyll was least soluble. That's why it got deposited here where the solvent kept rising. Chlorophyll B was more soluble. Chlorophyll A was even more soluble, and the most soluble of these was beta carotene because it traveled the longest distance. So a pigment that travels the longest distance, or the spot that travels the longest distance, is most soluble in that solvent. Then in B, you can see again the same kind of condition here, that there are further pigments separated, and again beta carotene being most soluble. And the solvent front is the line at which we allow the chromatography to stop. So this is the maximum point to which the solvent travel. So if RF value is to be calculated, so what is RF value? You divide the solute front by solvent front. So how do you calculate the RF value? You simply divide the solute and the solvent front together. How do you get these value? In the next part, they're asking you to calculate the RF value for beta carotene. So what will you do? You will find out the distance between the line of origin and the solvent front. You measure this and you get the solvent front, which means your value for how long the solvent traveled, and then the solute front from the beta carotene point to the line of origin. We always start to measure from the line of origin, and we measure it to the point where we are asked to measure. For example, if they're asking for beta carotene, we'll measure it for here. If they ask for chlorophyll A, we'll measure the point till chlorophyll A, and this will be our solute front. So the solute fronts are always measured from the line of origin to the point where the solute ended up being deposited. Whereas the solvent front is the measurement between this line and the line which is at the top, which means the whole distance traveled by the solvent. So you just apply the formula, you get the answer. The answer will not have any unit because it's a ratio. Then two explanations for the differences in the appearance of chromatogram A and B. Why A is having a shorter, you can say, thing there, because it was run for the lesser time. And then chlorophyll A changed to pheophylline. How can we say that? You can just compare this. So number one, the solvent line is different. Here the solvent traveled till the top, here the solvent traveled here. It couldn't travel up because we stopped this chromatography before the same time. Then we can see xanthophyll spot, we can see chlorophyll B spot, we can see beta carotene spot, but chlorophyll A has been further split into these two compounds. So these two pigments. So this is how when we use a separate solvent, the solubility changes and we get extra additional spots there. The student found some RF values for the chloroplast pigment such as spinach in a scientific paper. The values of in the scientific paper were different from the ones which are given here. Suggest one reason why this could be different. These values may differ because of the different solvents used in TLC. Always the solvents are the ones which are responsible for separating the pigments or the spots or the solutes. And in this case, they are the ones which lead to different values being there for this particular thing. Okay. Okay, I think I just there was a glitch there. Okay, let's move on. Now these are two kind of the spectra we have studied, action spectrum and the absorption spectrum. If you look at this spectrum, we can very well clearly see its absorption spectrum. Different pigments are shown to you and their absorptions are given to you. You can see the key there and how each wavelength is actually being absorbed or not being absorbed. So for instance, this area is not being absorbed at all by any pigment. And this is the area that gets reflected back. So they are saying compare the similarities and differences between the absorption spectra of chlorophyll A and carotenoids. So chlorophyll A is represented by this and carotenoid is represented by a dotted line. So we'll start to just compare these two things together. So this is the line for chlorophyll A. We'll just compare it with the carotenoids one, the dotted, and we'll just put together some values with the relevant units. We'll pick up the units where the trend changes and we'll put the values together comparative. Okay. When you are saying compare, so we are adding similarities as well as differences. So your marks will be split into both. So just don't include differences alone. You have to include similarities as well. In some species of plants, absorption spectra of light stimulates seed germination. The absorption of light increases the production of gibberellin in the embryo of the seed. Describe the role of gibberellin in germination of seed. Now, what was the role of gibberellin? Gibberellin is actually produced by the embryo. It goes to the allurin layer, it binds to the receptor. Once bound to the receptor, they form a complex with the enzyme, and then they cause destruction of GALA protein. This releases PIF. What was PIF? PIF is a transcription factor. This transcription factor, PIF, then attached to promoter and switch on the gene for transcription of amylase. The name of the gene is XET gene. The XEG gene gets activated. You don't have to remember the term XED, but at time in different MS, they allow that as a marking point. Amylase then enters the endosperm when it starts to hydrolyze the carbohydrate reserves which are stored there, such as starch and to maltose, and this breaks into its monomers, glucose, and also the embryos start to use the monosaccharides in respiration and growth. Come to question 4. Here they have given you a question of a wolf. Again, the first word in italics is the genus, and the second is the species. It lives in North America. Wolves may have gray or black coat color. The color of an individual wolf depends on its DNA, which it inherits, and the gene is mentioned. Wolves inherit two copies of this gene from each parent. Wolves that inherit one copy of black from this gene have a black coat. State the term used to describe an organism that has two copies of each gene. So two copies of each gene mean capital A, or capital A, small a, two copies of the same gene. This is a diploid organism. A form of a gene, allele. If you have a dominant allele or you have a recessive allele, so that's the allele here. Then a form of gene that gives phenotype effect or in a heterozygous, so dominant. Always it will give a phenotypic effect in the heterozygote as well, because for example, it's capital A, small a, capital A will still show its effect because it's a dominant allele. Okay. Then they're saying they are canine. Other than that, in addition to producing this black coat color, the protein coded for this gene also defends against infections, lung diseases. Canine distemper virus CDV causes serious lung disease in wolves. So a wolf can get like resistance against this infection if it's having that allele. Wolves have been previously infected with CDV, have antibodies against CDV, anti-CDV antibody. We know that any mammal when we get infected by an antigen, we produce antibodies against it, and the antibodies keep circulating in our blood for a long time, and the memory cells are there and all. We study that in AS. So the anti-CDV bodies will be there in the wolves that have been infected with the virus earlier. CDV can be passed from domestic dogs to wolves. This is where the transmission cycle occurs. That domestic dogs, they pass it on to wolves. Domestic dogs are more numerous in southern part of area occupied by wolves. Domestic dogs are less numerous in northern part areas occupied by wolves. So there are two areas occupied by wolves, the southern and the northern. Where are the domestic dogs more? Here you have more domestic dogs. Here you have less domestic dogs, although the wolves are still there in both. The relative frequency of black wolves compared to gray wolves increases from the north to south of the area they occupy. So the black ones, we know they are resistant to that infection, whereas the gray increases. They are saying why? So explain how natural selection caused the trend of distribution of black wolves. So here, the selection pressure is the infection. The pathogen is the selection pressure. The CDV acts as a selection pressure, and the allele black color gives the selective advantage, resistance against CDV. So this code gives a selective advantage. So dogs are common there. In South, more infection of CDV occurs in wolves, and black wolves have a selective advantage because they have the allele that confer advantage. They produce and they reproduce, they survive. When they survive, they are more likely to reproduce and pass on the allele to the offspring. This is increase the allele frequency of black coat. Similarly, you can give the reverse argument for the gray coat that this one is in more because the domestic dogs are lesser in that region. Okay, several different population of wolves were compared. This shows the relationship between the percentage of wolves population have anti-CDV antibodies and the percentage of black wolves. So you can see that this is a linear relationship. With reference to this, figure 4.1, state the relationship. You just state the relationship. And the percentage of the wolves anti-CDV body of wolf coat color, and suggest reasons for this relationship. So there's a direct relationship. As a percentage of anti-CDV increases, the percentage of black wolves increases as well. The wolves that survive, they will have the antibodies and they will pass on, and the gray wolf would have died. The black have the survival advantage there. So that's why the relationship just goes straight line. Next question. A simpler question in which they're asking about the endocrine system and the nervous system, and they're asking you to place tick and cross. So features of these three hormones: binds to receptors on the cell surface membrane. Yes, all three have receptors on the cell surface membrane. Results in the molecule moving from cells to blood. Yes, yes, and insulin, no, because insulin makes the glucose move into the cells, not into the blood. So that's why we'll press a cross there. Secreted by the result as a result of detection by osmoreceptors. Yes, ADH is affected by osmoreceptors, but glucagon and insulin are just because of the changes in the blood glucose level. So slower transmission in endocrine and faster in nervous system. Describe other ways in which they differ. So nervous system transmit through the neurons, where the endocrine system is not using neurons. They secrete hormones directly into the blood and transport it in blood. Electrical communication in the nervous system, whereas chemical communication in endocrine system. The effects of the nervous system are short-lived and more localized, while the endocrine systems are much longer and gives a widespread effect across the body. The endocrine system and the nervous system can affect the muscle function. So this muscle is shown to you. You can see that the sarcomeres are shown, the myofibrils here arranged. You can see the thick and thin filaments are there, the Z line, the M line. So you can see the lines here. You can see the sarcomere here. You can see that this part is the myosin, and this region, lighter one, is the actin. Okay, state the letter that indicates the length of a sarcomere. So the length of a sarcomere is from this disc to this disc between two Z discs, you can say, and this is the sarcomere. State the letter that indicates the region where actin and myosin overlap. So actin and myosin overlap in the region where you have more of this particular thing. For example, the region B, in which there will be an overlap of myosin and actin together. Explain the relationship between region D and changes during muscle contraction. So region D, where's region D? We know when the sarcomere shortens, this particular thing gets pulled together. So it's how the region D shortens when the calcium ions bind to troponin. We just mentioned the role of troponin here, how it changes the shape, and the tropomyosin then moves, and the binding site of actin and myosin gets like exposed, and they attach together. Myosin heads form the cross bridge, and to the exposed binding site on actin, forming a cross bridge. The power stroke occurs and pulling the myosin inwards, pulling the actin inwards towards the M line in the sarcomere. The overlap between actin and myosin, this continue to like increase. Why? Because more of the myosin will keep on pulling the actins together. Here you are given a list of question 6, a list of organisms and their conservation. You can say status is mentioned, and these represent the IUCN Red List, increasing risk of extinction, and the things here. Okay, this is a particular insect here. Explain how IUCN Red List assessments help to conserve biodiversity. So how it helps to conserve biodiversity here, how this particular thing affects. So what will be mentioned there? So for this, our main purpose will be if we go to the okay. So when we see how this thing goes on, just a moment. Okay, now this is gone. Explain how the IUCN Red List assessments helps to conserve biodiversity. So IUCN Red List identify the species that are at most risk for protection and can be prioritized. They collect the data and provide it to the government to take actions for protecting their species. This helps in conservation of that species and prevent from getting endangered. Because when you apply this list, you can see that obviously organisms which are EW or CR, they need to be prioritized. And where they need to be prioritized, whereas the organisms which are near threatened or least concerned, they will need lesser of the government resources or awareness because they already are in good number. Okay. With reference to figure 6.1 and the IUCN assessments of this suggests how the abundance of the butterfly differs across its distribution. So here we will just go for the condition which is mentioned here. The distribution of the large blue butterfly, this thing, again, this is genus and this is the species, extends across Europe and Asia. So you can find it all over the two continents. It is assessed by IUCN Conservation Nature, near threatened globally and endangered in Europe, which means that globally it is just near threatened, but in Europe it is endangered, which means it is more critical here. Or you can say there's an increased risk of extinction in Europe than globally. Okay, so this is what we'll just mention as they have asked. Then they are saying this has been successfully reintroduced into UK at 12 sites. These sites were restored to flower-rich grassland. So when you restore a habitat, you place you grow it in a way that the organism can thrive there. So the flower-rich grasslands were planted, and the conservation management actions designed for this thing to reestablish its colonies it at restored sites. These included 12 species of flowering plant, 8 other butterfly species, and 4 species of other insects. So all these were placed together. User information given to suggest why this P. arion went extinct in the UK. Why would it have gone extinct? Mainly due to habitat degradation. If How do we know that habitat degradation is the point of answer here? Simply because the conservation management was restoring the flower-rich grassland. If it's restoring the flower-rich grassland, that means the grassland were initially removed from that, and that must be due to habitat degradation, loss of habitats, either clearing above regions and using it for farming, urban development. This resulted in decrease in the plant flowers, and thus it affected the biodiversity. Outline the advantages of restoring habitats for endangered species. Restoration of the degraded habitats prevent extinction by allowing survival of the species there. This maintains the biodiversity. It can provide resource for recreational well-being activities as well as the scientific research. So let's move on. Question 7 deals with respiration. So outline the features of ATP that makes it suitable for universal energy currency. So we know ATP is a small water-soluble molecule that can move freely across the whole cell, throughout the whole cell. ATP hydrolyzes to ADP and phosphate group. This reaction results in releasing of energy. The ATP is an energy donor molecule. The reaction is reversible and ATP can be regenerated. Then they are giving you rotenone. Rotenone is often asked, although not in syllabus, but they give you the detail and then you can relate it very well. Rotenone disrupts the first carrier electron transport chain. So there are carriers in the electron transport chain. So the electron passes from here to the second, and then to the third, and then to the fourth, and eventually it falls into the matrix through the ATP synthase. And then in matrix, it combined with oxygen. So when it combined with oxygen, the electron transport chain ends because oxygen behaves as the final electron acceptor in aerobic respiration. So if rotenone is stopping the first electron carrier from working, so this is going to disrupt the whole ETC. And when the ETC will be disturbed, H plus ions will not go in the same way in the intermembrane space, and the whole thing will start to just seize up. Plus, reduced energy will not be oxidized further. So this is what we'll mention here. It disrupts the first electron carrier of electron transport chain. This carrier, the electron transport, this electron carrier is involved in oxidation and of reduced energy, regenerating and setting up the electron transport chain. Due to rotenone, ETC occurs less, so it provides less energy for protons to be pumped into the intermembrane space. This results in smaller proton gradient, less protons diffuse into the matrix through ATP synthase, fewer electrons reach matrix and end up in ETC. Energy cannot regenerate, so eventually glycolysis will stop because glycolysis cannot continue if NAD is not regenerated and it is not going back into the cytoplasm. Question 8 deals with a particular genetically modified soybean crop. It was grown in 1996, used in food products till 1998. It has been grown in 6 countries and used in food products in 21 countries. Table 8.1 summarizes the modification made by soybean to produce this particular GM crop. The gene PAT was taken from this particular organism, and the gene product is this particular enzyme. The function of the gene product, it stops the action of glufosinate herbicide. So glufosinate is the herbicide. We know the herbicides are used to kill weeds. So if you are growing this particular this genetically modified crop, you can freely spray glufosinate there. Glufosinate will keep on killing the weeds there without affecting your genetically modified crop. Why? Because it is having an alternate enzyme that keeps continuing the function, and it is not affected by the glufosinate. So this is what they are saying. So herbicide can still be used to kill weeds. Unwanted plants otherwise would compete with soybean because you have to remove the weeds from there, because if you do not remove them, they will eventually just use up the nutrition and from the soil and provide competition to the crop, reducing its yield. Name the type of organism that can be used to cut out, sorry, the type of enzyme. So whenever you are cutting the DNA, it's always cut by using restriction endonuclease. How you join the DNAs together? Using DNA ligase. Suggest reasons why this soybean is used in food products in 21 countries, but only grown in 6 countries. So as a product is there, but it is grown only in 6 countries. So these 21 countries may have unsuitable abiotic factors such as weather condition or soil minerals with no regulatory approval of GM crop, may have banned some of the group, may have opposed using of GM crops. There may not be enough land available, or the use of glufosinate may have been banned. So these are the possible reasons that why, although a country is using this particular soybean, but it is not growing in it on its own. A human kidney is shown in longitudinal section. X and Y are the regions. You know that X is pelvis and Y is medulla. So all these regions, these are the medulla, these ones, and this thing here is the pelvis. And if this was asked the outer part, so this entire is the cortex, and the outer part here is the capsule, and you find ureter somewhere here. Okay. A biosensor, outline how biosensor measures the concentration of glucose in urine. So outline, you just need to answer this in brevity. So we'll mention the name of the enzyme, glucose oxidase, which is immobilized on the strip. And this enzyme converts glucose to gluconolactone and hydrogen peroxide. The electrons produced by hydrogen peroxide, the quantity is proportional to the current being produced, and this current is then sensed by which in turn is proportional to the glucose concentration, and this is sensed by the electrode that amplifies the reading and displayed as a quantitative reading on the screen. Last part, last question, question 10. If explain the relationship between genes, proteins, and phenotype. This your syllabus demands you to relate the 3 things together: the gene, the protein, the phenotype, with reference to 2 examples of genetic diseases. So which gene, the name of the gene, the protein it codes, the phenotype it offers, and eventually if the disease is there, then that phenotype will be missing. So that's what they are asking you. Two examples. So we'll just recall the gene codes for a protein that results in the expression of a trait or a phenotype. The genetic diseases are an outcome of mutation in a gene that results in faulty protein and the results in disorder, the mutant allele of the gene F8. So we have named the gene. Codes for abnormal faulty factor 8. This results in hemophilia. So we have related the gene, the protein, and the trait. It is a sex-linked disorder caused by recessive allele of F8 gene if it codes for a faulty factor 8, so the blood does not clot. The gene locus is on X chromosome. Similarly, we move on to the second example, mutant HTT gene, the second example, because we have to give two examples here. Code for abnormal Huntington protein, we mentioned the name of the protein, similar to the one that we mentioned above here, the factor 8, resulting in the disease Huntington disease. The disease is Huntington and the protein name is Huntingtin. So do not confuse the two. It is caused by a faulty dominant allele. The gene locus is on a chromosome 4, not an autosome, which is an autosome. So we have used one example of a sex chromosome and the other as the autosome. So you can pick any. You can also give the example of the TYR gene for albinism or the sickle cell anemia gene. So either of them can be used. HBB gene. So these are the 4 genes that we have studied in our syllabus. So I have picked up these 2 for my answer. It is caused by the dominant. Okay, where this we just explained that what's the problem there. The CAG stutter occurred due to excessive repeated CAG triplet. The disease results in cognitive deficits and the movement difficulty and is fatal eventually. So this pretty much marks the end of your paper 4. I hope this video was helpful for you. That's it from my side. Thank you so much.